So, okay, chapter 12. This is the first PowerPoint of 3. First one's going to be the neuron and the myelin. So we're going to go over parts. We're going to go over what's myelin, and myelin for the rest of your life is just going to be insulation. So it's going to be that rubber cord on a an extension cord or something like that. Second one's going to be electrophysiology. Then we're going to look at how does the neuron transmit a signal, and then finally it's going to be the synapses. So the nervous system, obviously very complex. A lot of moving parts, a lot of different things that can be confusing. We need to have a good foundation of the nervous system to understand diseases. If you feel sore, or if you feel tired, or if you feel upset, you feel angry, it's good to know what's happening with your neurons, the ones in your brain, the ones to the muscle. Is it sensory? Is it motor? Knowing all these different parts in good detail can help you understand how you're feeling. So endocrine versus exocrine. AMP2, you're going to learn about endocrine. Right now, you're going to learn about nervous. But just to kind of give you some background, endocrine produces hormones. Those hormones go into blood. The nervous system, on the other hand, uses electrical signals and chemicals. Which one do you think is faster?
Nervous.
Yep, nervous, because it's just like turning a switch on or off. And endocrine would pretty much be like, oh, hey, can you turn the light switch off? It would take you a while to get over there. You would eventually do it, and then by the time you sit down, I say, hey, can you turn it off? Can you turn it back on? And you're going to be like, are you kidding me? I have to go back and turn it on? So because the endocrine is going to be using hormones, it's going to take some time for it to release the hormones, take some time for the hormones to travel to the target cell, and then to actually make some type of change. But nervous system, the one today, quick. So there's two basic subdivisions: central nervous system, peripheral nervous system. In your lab class, you're going to look at the brain and the spinal cord. Brain and spinal cord is going to be the central nervous system. Peripheral nervous system is everything outside of the central nervous system. So right when you have the spinal nerves going out into the muscles, or to the glands, or to the receptors, or to the sense organs, all of that's going to be in the peripheral nervous system. Now, the peripheral nervous system has two functional subdivisions. Sensory, which is the afferent division, and somatic sensory. So, okay, these are confusing. So you have a motor and you have a sensory. Afferent goes back to the brain. Motor exits the brain and goes back to a muscle or a gland. So if you see the word afferent or efferent, afferent's traveling back to the central nervous system, efferent is traveling away. And this is going to be tested on your exam, because a lot of the times you'll have these words afferent, efferent. Which part am I talking about? Motor or sensory? You also have something called the autonomic nervous system that has another name. You also have terms called sympathetic and parasympathetic. Have you all heard of that with the rest and digest or fight or flight? Knowing what all these individual parts of the nervous system do will help you understand the vocab of any of the test questions. So first, sensory afferent carries signals from various receptors to the central nervous system. It tells the central nervous system what's happening in the outside environment. If you're hot, you're cold, you feel pressure, something like that, it's too bright, all those are being brought to the central nervous system. Then the somatic sensory division carries signals from receptors, skin, muscle, bones, joints. The visceral, every time you hear the word viscera, it's going to be a membrane surrounding an organ. Those organs are always going to be in the middle, either thoracic or your pelvic or abdominal regions. So heart, lungs, stomach, and urinary bladder. This is going to be if you have stomach pains or something like that. They might not actually be stomach, they might be coming from the intestines. And that's going to be viscera, or those membranes that surround it that are going to send signals in. The other division, efferent, exits. So efferent signals are going to do something in the body. Most of the times I'll just say they go to muscles. Because if you wanted to walk away, or if you wanted to run, or you wanted to jump or skip around, you would be using the motor efferent division to do that. And just like the sensory, there's two divisions from that, somatic motor and visceral motor. The somatic motor carries signals to the muscle cells. So walking into a classroom, walking out of a classroom, you're using the motor division. That's a somatic motor division if you're using it correctly. The visceral motor division is called the autonomic nervous system. If you think the autonomic nervous system is complicated, I think chapter 15 in your book is dedicated to the autonomic nervous system. That's going to be one lecture in your next test. So the autonomic nervous system we kind of review, and all we're doing is we're going to take a closer look at sympathetic and parasympathetic. If you read the definition for sympathetic, tends to arouse the body for action, accelerating heartbeat, respiration, while inhibiting digestive and urinary. That's fight or flight. And if you've ever been had a rush of adrenaline, or you've ever been really excited, or you felt like you were going to jump out of a seat, that's your sympathetic division, where you're ready to run, all the blood's going to your muscles because you need those muscles to either fight or run away from danger. Parasympathetic, on the other hand, that's the one where you all feel how you eat a big meal. Think about Thanksgiving dinner. You kind of sit back and you relax, and you're like, okay, I can kind of close my eyes, I can kind of relax a little bit, because now you're going to have blood. They're going to be constricted towards the muscle, which blood is going to be shunted towards the digestive system and the urinary system, so you can start digesting food, and then the urinary system is going to eliminate the waste. So parasympathetic is your rest and digest. And yes, you can kind of memorize what each one does, but if you kind of remember fight or flight, on an exam, you're going to be like, yeah, when I'm ready to either run or do exercise or to get in a fight. Maybe you've never been in a fight, I don't know. But if you have, you feel all amped up on adrenaline and everything else. So your heart rate's going to go sky high. Your respiratory rate's going to start getting higher. So memorizing which is the autonomic nervous system, and it's the visceral motor, and then know what sympathetic does and what the parasympathetic does. And this is pretty basic, but central nervous system, brain, spinal cord, and here you can just see anything that leaves the spinal cord. We're going to look at those little bulges on the next chapter. Those are called dorsal root ganglia. Those start the peripheral nervous system. And here's a nice little chart of the central nervous system, the peripheral, where you have the sensory, visceral, somatic, motor, visceral is sympathetic and parasympathetic, and the somatic motor is just going to be muscles. This one's much more complex. So if you take a look at this one, you're like, oh my God, there's so many things going on. But if you kind of understand this chapter or this chart, then you'll fully understand your central nervous system and everything kind of going. We're going to learn about these receptors later on. We're not going to look at which ones. They're taking it from the external environment. This is going to be pressure and other stuff on the inside of the body. But pretty much, they're all going to be brought into the body. They're all going to go into the central nervous system, and they're going to interact with interneurons. Interneurons are just any neuron between the two parts. So sensory brings it in. The brain and the spinal cord has neurons that it synapses with. Those are called interneurons. Then it can either go to visceral motor, with somatic motor, and then from there, somatic motor goes directly to skeletal muscles, visceral motor, autonomic nervous system, either parasympathetic or sympathetic, fight or flight, rest and digest. So it kind of gives you a different way to look at it all, but much more complicated way to look at it. So, okay, what are the parts of the neuron? This is the easy part. So we're going to look at the 3 functional properties of all neurons. They have to be able to do these 3 things or they're not called neurons. We're then going to look at the basic functional categories of neurons. We're going to look at the parts of the neurons, because if you understand the parts of the neuron, you understand how the neuron works. And then at the end of this lecture, we're going to look at how does a neuron transport materials between the cell body and the tip of its axons. And if you know anything about Parkinson's or Alzheimer's, those are problems with the transportation of material. Stuff builds up, like in Alzheimer's, beta amyloid plaques will build up, and they'll kind of stop the ability for the axon to work properly. So what are the 3 things that they got to do? Excitability. They have to be able to turn on or turn off. So every single neuron has to respond to some type of change. Next, it has to be able to conduct that signal. So it wouldn't make any sense if they can get excited, but then nothing happens. So they're going to have something called an axon that travels and transports that signal to something at a distant location. Distant location can be a couple nanometers, or if it's going all the way to your toes, it can be about 1 meter long. Then it has to secrete. It has to be able to talk to that target cell. So not only does it have to reach that target cell, it has to be able to talk to it. And it does that by releasing a neurotransmitter. So every single neuron has to be able to be excited, conduct something, and then secrete a neurotransmitter. You have 3 general classes of neurons: sensory, to the central nervous system we talked about. Interneurons we talked about. They're the ones that communicate between the sensory and the motor. So nothing new. And here's just sensory, interneuron, and then the motor. And because they're in red, there's a good chance it's going to show up on the test. What are the 3 types of neurons? So first, the cell body. So structure of a neuron, the cell body or the soma is right there. Sometimes it's called neural soma, cell body, but you'll usually see soma. If you have different lab teachers, they probably call them different things, but everything right there, the cell body. So everything you learned in the cell in what is it, chapter 2 or no, chapter 3, it has a mitochondria, it has Golgi apparatuses, it's got rough ER, it's got all of the same functions that a normal cell does, but all of them are going to be called the soma. It has one nucleus and one nucleolus, just like most cells do, except for muscle cells. And then the next thing are these little hair-like looking things called dendrites. Dendrites are what carries the signals into the soma. And if you're studying neuroscience and you want to get into more detail with what is a neuron and how do they work, we talked about they have to be able to talk to each other. They're either going to send an excitatory or an inhibitory response. What type of responses can come in via dendrites? So the dendrite's the primary site for receiving signals. The more dendrite a neuron has, the more information it can receive, and then incorporate that into the decision making. The axon is the nerve fiber that leaves the soma. You have a little swelling at the end called the axon hillock. The axon hillock is where those competing forces kind of come. If it's excitatory or inhibitory, they all kind of go down to the axon hillock. And if it reaches threshold, it fires down the axon. And that's why once you have the impulse, It's called an all or none impulse because if it fires down the axon, you can't bring it back up. You can have collaterals on an axon, which just means the axon goes in two different ways. And then at the end of the axon, you have these little synaptic knobs. And then you kind of see these little, I don't know, they look like little hot dogs on the axon. Those little pills are little nodes or sections of myelin sheath. And the peripheral nervous system, it's the Schwann cell that's going to maintain the myelin sheath. So Schwann cell, myelin sheath, Schwann cell, myelin sheath. That's what makes that an internode. And then in between the nodes, you have a node of Renvier, an internode, node of Renvier, and then each one of those has myelin. And when we talk about how the signal jumps down the axon, the myelin and the node of Renvier is going to become important. Get down to the end. Synaptic knobs. Synaptic knobs are going to have neurotransmitter. In your lab class and in this class or the last test, you learned about acetylcholine because acetylcholine works on nearly every single muscle cell in your body. So acetylcholine is what we're going to use here. These are called synaptic knobs, synaptic boutons. I've had teachers call them that. You can call them synaptic buttons, terminal buttons, or terminal boutons. I don't know why they keep trying to make this word sound more interesting, but it looks like button, it's spelled button. But if you have a teacher or if you go to a neuroscience conference, you're going to hear this called a bouton, and I still think it's kind of funny. But that's where one neuron is going to talk to the target cell, or it's going to talk to another neuron. You can have multipolar, which means, they have different poles. They can receive a lot of information. You can have bipolar, which if you're not looking at it in tissue, you don't know which one's the dendrite, which one's the axon. Unipolar, where it looks like the axon is actually the dendrite, or anaxonic, which can be in the brain. Those are going to be horizontal cells. Oh, sorry, the retina. I should know this. Anaxonic. They do have an axon in a way, but all they do is they receive a bunch of information and then they release stuff to neighboring cells to help talk to them. And they're going to be in the retina. So, okay, what are the supporting cells? The neural glia. Every time you see the word glia or neural glia, these are just going to be supporting cells. Some of these are going to be extremely, extremely important in the future. So if you learn them and hear about them on the news or in reading or in research, this is where a lot of science and money for science and research is going. There are 6 types of cells. We're going to look at a couple of them because a couple of them are very important and some of them aren't as important yet. We're going to describe the myelin sheath. We're going to talk about 2 types, oligodendrocytes and Schwann cells again. Those are type of glial cells. And then we're going to look at unmyelinated nerve fiber versus a myelinated nerve fiber and what happens to the supportive cells. And then what happens about damaged nerve cells and how they grow. So about 1 trillion neurons and about 50 to 1 neuroglia for it. Neuroglia are just supportive, helpers. They either help the neuron directly by providing assistance or nutrients. It either provides a better environment for the neuron. It cleans the environment around the neuron. But in other words, it's very helpful to a neuron. Without the neuroglia, neurons die. So they do a bunch of different things that help the neuron. You have an oligodendrocyte. You're going to see this later on in this lecture. Oligodendrocytes provide the myelin sheath in the central nervous system. If you think about the central nervous system, all the neurons are pretty close together. So one cell can then provide myelin for multiple axons that are close to it. So that's why in the central nervous system you have an oligodendrocyte. The one we just talked about was a Schwann cell. That's in the peripheral nervous system. Epidermal cells. Very, very, very important. They line internal cavities of the brain. They're cuboid epithelium with cilia, with little kind of feet on their top surface. They secrete and circulate cerebral spinal fluid, and the cerebral spinal fluid is that clear liquid that bathes the brain with nutrients and helps keep it buoyant. We'll talk about that with the brain. But epidermal cells, extremely, extremely important for that blood-brain barrier, but we'll cover that later. Microglia, it's not in red. It's not in red because they're small and they don't know a bunch about them. They are parts of white blood cells called the monocyte that kind of migrate into the neurons or next to the neurons. And what they do is they phagocytose debris or other organelles and other things. The one I have down here, astrocyte. Astrocytes also are very important for the blood-brain barrier. They also are the most abundant cells in the central nervous system. And what they do is they have these little feet. Those feet will go and kind of grab on, if you kind of picture this line of words as just a capillary, their feet will start to hold on to the outside of the capillary and start to create tight junctions, which means that stuff cannot pass through them. They have to go through these little holes. That's how your blood-brain barrier is formed, and it's formed by these astrocytes. They also have a lot of different other functions that they don't even know what they do. In the rest of the body, there's astrocytes. Those astrocytes are always going to be around capillaries and blood vessels. They might help the growth of blood vessels. They might help providing the pathway for blood vessels. So if you see astrocytes in the future, they're doing a lot of research into what astrocytes are actually doing in the brain. And because if you think about cancer, it's just the growth of blood vessels that shouldn't be there. Stroke, you need to have blood vessels back to those damaged regions. So that's why they're looking at astrocytes in cancer and stroke and other types of issues. Peripheral nervous system, we talked about Schwann cells, so I don't want to repeat myself, but myelin sheath. Satellite cells, these are going to be around the neurosomas and ganglia of the peripheral nervous system. And pretty much what the satellite cells do, they also have feet, and they're just going to provide kind of some insulation on the soma so signals can travel all the way down to the axon terminus or axon hillock. And there's your astrocyte and its little feet, and you can see how they're creating little holes in that capillary, and that's the whole basis of your blood-brain barrier. And there's your oligodendrocyte, providing myelin on multiple axons. And if you say oligodendrocyte, it's almost like you have marbles in your mouth. Try to say it. It's fun. Or not. Oligodendrocyte. So what's a glioma? What's a tumor? Because neurons don't grow a lot, usually neurons are not going to be the source of tumors. So tumors usually come from someplace else. And a tumor in the brain or a tumor in your muscle is just going to be rapidly dividing cells. Something in their DNA shuts off the kill switch, so they just keep growing and growing and growing until they destroy the tissue that it's in. And neurons, they don't really grow a lot. So meninges, the dura mater, the arachnoid matter, the pia mater, they grow. So cancer and tumors can start coming from these meninges. Also, something other part of the body, it can metastasize, go into the brain tissue, get lodged in there, usually through the blood supply, and then those are going to be tumors in the central nervous system just because they came from someplace else, and then they go into the brain. And then sometimes glial cells are another source of tumors. And when you have a glioma, They grow rapidly, very highly malignant, which means they break off and go to different parts of the brain. And instead of having just one tumor, you have multiple tumors all throughout the brain tissue. You can have the blood-brain barrier decreases effectiveness of chemotherapy. The reason why the blood-brain barrier doesn't really work with chemotherapy, all chemotherapy drugs, except for one. It's a very long molecule. It's not that long, but it's too big to fit through the small holes of the blood-brain barrier. So they have to use special chemotherapy drugs that aren't as efficient. So what they do with gliomas and tumors, radiation or surgery, or sometimes surgery with radiation. That's when you have to know about alpha and beta particles. Sometimes they can put an alpha or beta particle in the tumor for a little bit and then remove it through a straw or a tube. So the myelin sheath, insulation. Hopefully you know about that by now. If you have the myelin sheath, it moves a lot quicker. Myelination begins at week 14 of fetal development. You might think, oh, wow, before that it didn't need myelin. The cells were just too close together at that point, so there's no point. The neurons can actually send some signals before 14 weeks, not really much, but because they're so close together, unmyelinated nerve fibers can still talk to each other. And then they proceed rapidly, obviously, and then throughout your life, you're always remyelinating your cells. But dietary fat is important for myelin. The Schwann cell is going to spiral around the axon and provide the myelin sheath. Then there's going to be a node between them called the node of Ranvier. Oligodendrocytes do that process in the central nervous system. Many Schwann cells or oligodendrocytes are needed to cover one fiber. They're segmented between the internodes we talked about and the nodes of Ranvier. Those are going to become very important on how the signal gets moved down the axon. There's your oligodendrocyte again. There's your Schwann cell and how it wraps around the nucleus. Multiple sclerosis. And we're going to talk about Tay-Sachs disease after this. But multiple sclerosis, the oligodendrocytes and myelin sheath in the central nervous system start to break down. Instead of just replacing it with more myelin, it gets wrapped around in scar tissue. Because scar tissue is not high in lipids, it's high in connective tissue, the nerve impulse can't pass through the axon. It doesn't skip, it just kind of stops. So if you have a wire that just sends signals every once in a while whenever it wants to, or whenever the charge builds up so much that it sends it through, you can start looking at this and saying double vision, tremors, numbness, speech defects. This is when you're trying to do something but you can't because that impulse from the nerve kind of gets blocked. So you start to shudder, you start to kind of shake, you start doing all these things. Like if it's in the optic nerve will create double vision, and because it's in the central nervous system, the optic nerve is close to the brain, so sometimes that gets damaged. It usually begins around 20 to 40 years of age, and then about 20 to 25 to 30 years after that, people died. The reasoning is they don't know what causes it. It might be a virus, and then the virus might trigger some type of autoimmune disease. But what happens, too much scar tissue builds up over those 25 and 30 years, that nerve impulse for breathing, thought, different types of behaviors that we need to survive, they just can't process anymore, so patients will die. Tay-Sachs, on the other hand, is just as bad, but it's going to be a hereditary disorder of infants of Eastern European Jewish ancestry. And it is caused by an abnormal accumulation of glycolipid called GM2. And I'm not even going to try to say it. It's this one down here. But But GM2 builds up in the myelin sheath. You have an enzyme that destroys it. If you have that enzyme to destroy it, no problem, you're healthy. But in Tay-Sachs, because it's a hereditary disorder, you don't have the gene to make this lysosomal enzyme. If you don't have that lysosomal enzyme, that means this GM2 starts to build up. Once it builds up, it stops the conductive of nerve signals, blindness, loss of coordination, dementia, usually fatal before the age of 4. And the reason why it's fatal before the age of 4, it builds up, it builds up, it builds up, and then eventually can't get rid of it. So by the age of 4, so much is built up into the body that all of those processes start to break down again. And there's your unmyelinated axon with a myelinated axon. You can see how thick the myelin sheath is. And every time you see myelin, it's just insulating the axon. And then what creates the fastest axon? First, it has to be insulated. So it has to have myelin. Then the diameter changes. So if it's a large diameter, it's quicker. Smaller diameter, it's slower. So if it has a very large fiber, it can conduct very, very fast. If it has myelin, it conducts it even faster. So on your test, you might have a question like, I'll give an example of a large diameter with myelin. On this chart, you can see it moves at about 120 meters per second. Small unmyelinated fibers, about 1/2 to 2 meters per second. A small diameter axon with myelinated, 3 to 15 meters per second. Think about how long it takes you to wiggle your toes. You can wiggle your toes almost immediately. It's because that signal is traveling from the brain via a large diameter that's myelinated. And that's it.